Hello there, let's look at Mendel's first law of inheritance, which is the law of segregation. So the law of segregation states that members of each pair of alleles separate or segregate during the formation of gametes. The member of alleles, remember alleles comes in pairs. We inherit one from the parent, from each parent, and then we come up with a pair of alleles. But this pair during gamete formation, they separate and segregate. That's what the law of segregation states. So only one allele can be carried in a single gamete. So when gametes are formed during meiosis, only one allele can be carried in each gamete. So one allele for each trait is inherited from each parent to give a total of 2 alleles. So this law of segregation can be illustrated by what we call a monohybrid cross, which illustrates the inheritance of only one trait. So a monohybrid is a cross which illustrates or shows how one trait is passed on from parents to offspring. For example, let's look at the monohybrid cross between a dominant round shaped seed and a wrinkled shaped seed. In this case, we are going to say a dominant round shaped seed is homozygous, which is it has all the alleles are dominant, and a wrinkled seed, let's say it has recessive. So for a wrinkled seed, for a seed to be wrinkled, then it must have recessive alleles. So recessive alleles, these are alleles which cannot express themselves in the presence of a dominant allele. So to draw a monohybrid cross, you first identify the trait involved in the cross. So the trait we want to see here is the seed shape. The seed shape is the trait, and the alleles involved, we have the dominant R and the recessive R, which represents wrinkled. And then for a cross, we are going to cross the round seeds with a wrinkled seed, and the genotype are homozygous round and heterozygous wrinkled. So we are going firstly to use a Punnett square, then from there we are going to use a genetic diagram to see how the traits are passed on in a monohybrid cross. So for a Punnett square, you draw a square like this one here, and you divide it into 4 equal boxes. Then from there, get the genotype of the first parent and write them on the separate boxes like this at the left, and then from there on the top write the alleles for the other organism or the other parent there. So you write them there. After that, now match these alleles R with this R here, then write the result there. So we're going to have capital letter R and small letter R in this first square, and then for this one with that one we are going to have the same result, and then this one with this allele here we are going to have that result there, and this one with that again we are going to have these results there. So you can see that we all have the same type of the same genotype, meaning all these offsprings are going to be round. Why? Because this capital letter R is dominant, meaning a recessive, which is small letter r, cannot express itself when a dominant allele is present. So all these offsprings are going to be round shaped seed. So we're going to we have the genotype, which is heterozygous, capital letter R and small letter r, and the phenotype is round. So the genotype ratio, all of these seeds are alike, they have the same genotype, and for the phenotype we also have the same kind of phenotype. So remember, a phenotype is simply a physical appearance of the offsprings. So these offsprings are all alike because they have the same genotype. This information can be illustrated using a genetic diagram. So for a genetic diagram, we don't draw a Punnett square, what we do, we identify the trait first, which is in this example is seed shape. We identify the parent phenotype. The appearance of the parent, we said it is round shape crossed with the wrinkled shape. Then the genotype of the parent. The genotype is the genetic makeup of the parent. For the round seed, we have homozygous dominant, so it is capital letter R and capital letter R. For wrinkled, it is recessive, so we are going to represent it with small letter R. So during meiosis, which is during the segregation, these alleles are going to separate, and then we are going to have gametes which have half of the genotype of the parent. So we're going to have capital letter R and capital letter R separately, and then for this we are going to have small letter R and small letter R there. So you cross this during fertilization. For the first R, with this R here, we are going to have this, and if this again with that other R, which is that other allele, which is that side here, we are going to have this offspring here, and for this one we are going to have these offspring there. That we call this the F1 genotype, or the genetic makeup of the F1 generation. These are offsprings resulting from the P1 parents, those P1 parents there. So the ratios, for the ratios or the phenotype, the F1 phenotype, all of these are going to be round because we have the dominant R, which is the round shape there, which is dominant. So the genotypic ratio, we have all have the same genotype, which is capital letter R and small letter r, and the phenotype all are round. So this is the genetic diagram, and this is a Punnett square. So it is the same way of illustrating a monohybrid cross. Okay, so let's cross these offsprings here. We see what we are going to get. So if you cross the offspring, we are going to use the same method where we identify the trait. The trait in this case, it is the seed shape. Alleles, we have capital letter R, which is for round, and small letter r, which is for wrinkled. So the cross is in between the round seeds and the other round seed, because these offsprings here are all round. So we are crossing these offsprings here. So we have the genotype, which is heterozygous for all offsprings, capital letter R and small letter r, capital letter R and small letter r. So what you do, you write these alleles separately here using a Punnett square. You write this one this side and that one there, and this other one you put it on top there as the way we have done there. Then from there, during crossing, this R here is going to cross with that, and we are going to have all dominant homozygous alleles. And then for this one and that one there, we are going to have heterozygous offspring, and this one with this allele here, we are going to have a heterozygous allele, and this one lastly with that allele there, we're going to have a wrinkled seed. So this is what Mendel did, and he found that these other offsprings, the three, are all round, because in here it is homozygous dominant and here it is heterozygous, meaning these are round and that one is round. But for this one, since we do not have a dominant round allele here, then it is a wrinkled seed. So we found all the three were round, but the other one is wrinkled. So the genotype, we have this dominant homozygous, and we have heterozygous, these two here, and then we have a wrinkled seed there. So for the phenotype, we are going to have round, which is these three are round shaped seeds, and one is wrinkled. Now the ratio, for the ratio, the genotypic ratio, we are going to have 1, which is homozygous dominant, and 2 are heterozygous dominant, and 1 is wrinkled. So we show that there. And then from there, the phenotypic ratio, the phenotype, the outside appearance, how these seeds are looking like. So the appearance of these three seeds here are all round. So we are going to have 3 to 1 wrinkled seed. So this information can also be illustrated using a genetic diagram here. So genetic diagram, you follow the same steps, but in this case now we do not draw a Punnett square, but we just separate them here and show all the necessary steps like I showed you in the first cross. So what can we summarize about the monohybrid cross? So a monohybrid cross involves what we call a pure breeding parents. So in this case, we were crossing a pure breeding round seeds, which carried capital letter R, which is homozygous dominant. Then these seeds are said to be homozygous dominant, while the pure wrinkled seeds are said to be homozygous recessive. And then from there, the alleles exist in pairs, but during meiosis they separate into during meiosis 1 of the 2 alleles is transmitted separately to each gamete. So alleles exist in pairs, but during meiosis they separate and are transmitted separately to each gamete. So each gamete will have 1 allele. Then from there, during formation of F1 hybrids, different alleles are brought together but do not contaminate. When you are crossing those crosses, we did not see where these alleles were being contaminated. So these alleles come together, but they do not contaminate each other or mix up as they get separated when forming the F1 gametes. Okay, so this is what we can summarize about a monohybrid cross. So thank you very much for watching. Don't forget to subscribe. Leave out your comment concerning this video in the comment section below.